Dear students, very warm welcome to IG Academy. Now we are going to discuss on the topic reproduction in organisms. All living organisms in the earth, let us say in the galaxy, which shows a life cycle that involves birth, growth, development, maturation, reproduction and death. Every organism follows the same introduction. Reproduction is a biological process by which organisms produce their young ones, and it is considered as a fundamental feature of all living organisms. The young ones grow and mature to repeat the process. Reproduction results in the continuation of species. Both asexual and sexual reproduction have some basic features that includes synthesis of RNA and protein, replication of DNA, growth, cell division, and formation of reproductive units. There are two modes of reproduction. One is asexual reproduction and the other one is sexual reproduction. Asexual reproduction. Here, only one parent is required. Since there is only one parent, there is no fusion of gametes and no mixing of genetic information, usually by amitotic or mitotic division of somatic cells. Hence, it is called as somatic or blastogenic reproduction. As a result, the offsprings are genetically identical to the parent and to each other. Many organisms will have asexual mode of reproduction, and it is more common among lower groups of organisms like Protista, bacteria, archaea, and multicellular animals with simple organization. As I said earlier, here in asexual reproduction, the offspring shows uniparental inheritance without any genetic variation. Different modes of asexual reproduction in animal are fission, sporulation, budding, gemule formation, fragmentation, and regeneration. And this is the skeleton of the types of fission. There are four types of fission seen in animals are binary fission, multiple fission, strobilation and sporulation. Based on the plane of division, binary fission, which is further divided into four types: simple irregular binary fission, transverse binary fission, longitudinal binary fission and oblique binary fission. In shortcut, let us have the types of fission with example. For binary fission, example is Euglena, and for multiple fission, it's Plasmodium. For sporulation, it is Amoeba, and for strobilation, it is Aurelia. So fission. The parent body divides into two or more identical daughter individuals. There are four types of fission seen in animals, as we discussed earlier in the skeleton. So first one is binary fission. The parent organisms divides into two halves and each half form a daughter individual. Initially, the nucleus divides amitotically or mitotically, followed by the division of cytoplasm. The division of nucleus is called as karyokinesis, and the division of cytoplasm is called as cytokinesis. The resultant offsprings are genetically identical to the parent. We can see binary fission in the form of diagrammatical representation. The parent cell, first the nucleus divides by mitotic division into two, and then the cytoplasm division occurs. Here we can see the fission point, and then finally this divides into two new daughter cells. Types of binary fission, which is simple irregular binary fission, transverse binary fission, longitudinal binary fission, and oblique binary fission. All these types are based on the plane of fission. Simple irregular binary fission, which is seen in irregular shaped organism. Let us take amoeba, which does not have any regular shape. The nucleoli disintegrate and the nucleus divides mitotically. The cell constricts in the middle, so that cytoplasm divides and forms two individuals. Let us see in the diagram. Here first the nucleus division occurs, and then the cytoplasm divides, and finally this forms a two-daughter amoeba. The second one is transverse binary fission. The plane of division is along the transverse axis of the individual. Example, paramecium and planaria. In paramecium, the macronucleus divides by A mitosis and the micronucleus divides by mitosis. So this is the diagrammatic representation. Here we have micronucleus and macronucleus. The micronucleus divides mitotically and the macronucleus divides amitotically. Again, the plane of division is transverse. Finally, two new daughter paramecia was formed. The third one is longitudinal binary fission. The nucleus and the cytoplasm divides in the longitudinal axis of the organism. Example, Verticella and Euglena. So this is Verticella, and in Euglena, first the nucleus divides into two, and then we can see the longitudinal furrow that occurs from top to bottom. Finally, two daughter new Euglena was formed. Final type is oblique binary fission. Here the plane of division is oblique, which is seen in dinoflagellates. Example, Serratium. So this is the fission point. And then the second type is multiple fission. The parent body divides into many similar daughter cells simultaneously. First, the nucleus divides repeatedly. Later, the cytoplasm divides into as many parts as that of nuclei. Each cytoplasmic part encircles one daughter nuclei. This results in the formation of many individuals from a single parent organisms. This is called as repeated fission. Example, Verticella. So multiple fission in Plasmodium. Here in Plasmodium, multiple fission occurs in schizont and in oocyte stages. The multiple fission in schizont is called as schizogony, which produce merozoites. Multiple fission in oocytes is called as sporogony, which produces sporozoites. So let us see in the diagram. Schizont contain the single nuclei. Then the nucleus undergoes division and forms a number of daughter nuclei, and each forms into merozoites. The schizont wall ruptures when the merozoites emerges out and they develop into an individual young ones. Multiple fission in encysted amoeba occurs during adverse condition. Withdraw pseudopodia and forms a three-layered cyst wall that is called encysement. Under favorable condition, the multiple fission producing pseudopodiospores. After maturity, the cyst wall breaks and liberates the pseudopodiospores. So let us see in the diagram. We can have this three-layered cyst wall and the formation of daughter nuclei, and then the pseudopodiospores formation. Finally, under favorable condition, the wall ruptures and the young amoeba which is formed. The third type is strobilation. In strobilation, several transverse fission occurs simultaneously, giving rise to the number of individuals which does not separate immediately from each other. Example, Aurelia. And this is the diagrammatic representation. Plasmotomy. It is a type of fission which is seen in multinucleate organisms. In plasmotomy, the multinucleate parent along with the nuclei divides into two or more daughter individuals. So each new individual has many nuclei but not equal to the number of parent organisms. After fission, the nuclei of the daughter cells undergoes to equalize the normal number. Example, Opalina and Phleomixa. So this is the diagram. The next one is sporulation, which is seen in amoeba. During unfavorable condition, first the amoeba multiplies by sporulation and the nucleus breaks into small fragments. Each fragment develops a nuclear membrane, becomes surrounded by the cytoplasm and develops a spore case. When the condition becomes favorable, the spores are liberated and each hatching into a young amoeba. So this is a diagram. Budding. Budding is of three types. One is endogenous budding and exogenous budding and gemmules. Budding. The parent body produces one or more buds and each grows into a young one. After complete development, the buds separate from the parent body and lead an independent life. Example, hydra and sponges. So this is budding in sponges. Initially, the individual parent, in that the bud forms and grows and separates and leads a new individual life. Second one is budding in hydra. So the growth of the bud, and after growth, mouth and tentacles will be formed. Finally, it gets detached from the parent and leads an independent life. The types, the first one is exogenous budding. The buds are formed on the outer surface of the parent. Example, hydra. When the food is in plenty, the ectoderm cells increases and forms a small elevation on the body surface. The buds enlarge, develops mouth and tentacles. When fully grown, separate from the parent body and leads an independent life. So this is the diagrammatic representation. Initially, the bud forms and then the growth of tentacles and mouth, and after full development, bud detaches from the parent and becomes an independent hydra. Endogenous budding. Buds are formed inside the cytoplasm and many remains within the body of the parents. Example, Noctiluca. So here all the buds which remains inside the parents. Gemmules. This is a peculiar mode of asexual reproduction by internal budding called gemmules. Example for this is freshwater sponges and in some marine sponges. During unfavorable conditions, the sponges disintegrate, but the gemmules can withstand the adverse condition. The adverse condition may be like temperature is high or it's very low, or if there is a deficiency of food, there may be any reason. So gemule, which is a hard ball having a mass of food-laden archaeocytes. When the condition becomes favorable, the gemule begins to hatch. So this is a diagrammatic representation of a gemule, which is a hard ball and having a mass of food. Fragmentation. Here the parent body breaks into fragments. Each fragment develops into a new individuals. Example, sea anemone and planaria. We can see in planaria, if the parent planaria, which is cut into fragments, and each piece has an ability to develop into a new daughter planaria. Sea anemone also have the same feature. Fragmentation in sea anemone. Fragmentation in sea anemone is referred as pedal laceration. When the sea anemone gets detached and moves, the pedal disc breaks off. Lobes are constricted from the leftover pedal disc and each lobe develops into an entire organism. So this is the diagrammatic representation. Here this is the pedal disc. Apolysis. In tapeworm that needs two hosts to complete their life cycle, the first host is human being and the other one is pig. So in tapeworm, the gravid proglottids, that is in the posterior end, that is which is a matured one, are regularly cut off either singly or in groups from the posterior end. This shedding of gravid proglottids is called as apolysis. These proglottids, which contains embryos and on entering into the host, say it's considered as a pig, and it develops into a new individuals. The embryo has a capacity to develop into a new individuals when it enters into the host. Regeneration. Regeneration is regrowth in the injured region. It was first studied in Hydra by Abraham Tremblay in 1740. Regeneration is of two types. First one is morphallaxis and the other one is epimorpholosis. In morphallaxis, the whole body grows from a small fragment. Example, Hydra and planaria. When Hydra is accidentally cut into several pieces, each piece can regenerate the last parts and develop into a whole new individuals. The same case is for planaria also. Epimorphosis. It is the replacement of lost body parts. It is of two types. One is reparative and the other one is restorative. So in reparative, only certain damaged tissues can be regenerated. For example, damaged liver tissues. In restorative, several body parts can develop. Example, tail of lizard and arms of a starfish. So unfortunately, if the tail of the lizard was cut off, it has the ability to develop its lost parts. Same case is for starfish. Starfish also has the ability to regenerate its lost arms. So that's it for the today's session. We can have the discussion on sexual reproduction in our next video. Thank you.